Good afternoon everyone. In the last session we have studied about the functions of kidney. Now we will study the structural and functional unit of kidney. What is the structural and functional unit of kidney? It is nephron. Okay. So in each kidney total 1 million nephrons are there, and as the age increases nephrons may decrease in number but nephrons cannot increase in number. Okay, जैसे कि neurons cannot divide and multiply. Okay, similar way nephrons in the kidney cannot divide and multiply. So उनका number increase नहीं होता है after birth, उनका number decrease हो सकता है. Got it? So how much amount of nephrons are present in the kidney? One kidney has 1 million nephrons. Okay. Now in this particular session we'll study in detail structure of nephron. Okay. So what is nephron? Nephron is the structural and functional unit of kidney. Okay. So last time जो हम लोग ने किडनी का फंक्शन स्टडी किया था उसमें मैंने बोला था मेन function of kidney is elimination of nitrogenous waste, that is urea and uric acid, followed by osmoregulation, that is water and salt balance is also maintained, and it is all possible because of the nephrons which are present in the kidney. Okay. So now let's study the structure of nephron. So this is the diagram which I have drawn about nephron. Okay. So each nephron which is there, it consists of which all parts? Okay. What is this? This is the Malpighian body. Malpighian body which is there, it consists of Bowman's capsule and glomerulus. So very first part of nephron is Malpighian body. Malpighian body is made up of this cup-shaped structure which is known as Bowman's capsule, and in this cup-shaped structure there is a network of blood capillaries, and this network of blood capillaries is formed because of entry of afferent arteriole in this cup-shaped structure. Okay. What is this afferent arteriole? Afferent arteriole is one of the branch of renal artery. I have told you when we have studied less of kidney, I have told you renal artery enters in the kidney. Renal artery जब भी enter होता है kidney में, it divides into many interlobal arteries. Okay, and these interlobal arteries which are there, these will further divide and redivide into many arterioles, afferent arterioles, and each afferent arteriole will enter in different nephrons. Okay, as I told you millions of nephrons are present in the kidney. So हर एक nephron का जो Bowman's capsule में जो cavity रहता है, Bowman capsule encloses cavity, and in this cavity this afferent arteriole which is branch of the renal artery will enter and divide and redivide into many blood capillaries, and this network of blood capillary is known as glomerulus. Okay, and once the many division of the blood capillaries takes place, these blood capillaries will then unite to form efferent arteriole. So now look at the diameter of afferent arteriole and efferent arteriole. किसका डायमीटर ज़्यादा है? एफरेंट का एज कम्पेयर टू इफरेंट आर्टेरियोल. ओके, सो एफरेंट आर्टेरियोल का डायमीटर ज़्यादा है तो उससे ब्लड ज़्यादा जाएगा. गॉट इट? सो प्रेशर blood pressure which is present in the afferent arteriole is little bit lower as compared to blood pressure which is present in the efferent arteriole. Okay, so efferent arteriole में blood pressure why it will increase? Because afferent में जो blood pressure है वो कम है, volume of the blood which is entering is more. Okay, so blood जो जा रहा है blood capillaries में amount of blood is more, but when the blood capillaries which are there, these are uniting and these unite to form efferent arteriole. So efferent arteriole का diameter कम है, volume of the blood will be less in the efferent arteriole as compared to the volume of the blood which is present in the afferent arteriole. So blood pressure will increase in the efferent arteriole as compared to blood pressure which is present in the afferent arteriole. Got it? समझ में आया? So basically we have studied two parts of the nephron. Malpighian body which is there, it consists of Bowman's capsule and glomerulus. Bowman's capsule is cup-shaped structure and glomerulus is the network of blood capillaries which is formed due to division and redivision of the afferent arteriole. Okay, and these blood capillaries will reunite to form efferent arteriole. And this efferent arteriole will come out from the Bowman's capsule and it will divide and redivide around the nephron. Efferent arteriole will divide and redivide into peritubular capillaries. Okay, and these peritubular capillaries which are there, these are around the nephron. Peri means around, tubular मीन्स नेफ्रॉन के ट्यूब्यूल्स के आजू बाजू में ये कैपिलरीज है फैले हुए. सो दीज कैपिलरीज विच आर देयर अराउंड द नेफ्रॉन, ट्यूब्यूल्स ऑफ द नेफ्रॉन, इट इज बिकॉज ऑफ डिविजन ऑफ द efferent arteriole. Got it? So very first part of the nephron, is it clear to all of you? Malpighian body which consists of Bowman's capsule and glomerulus. Now we'll talk about the next part of the nephron. Okay, next part of the nephron, how it is? It is tubular. Whole part of the nephron is tubular, tube जैसा है. So Bowman's capsule is leading to the renal tubule. Okay, so renal tubule is the other part of the nephron. Nephron renal tubule which is there, it is having three parts. So renal tubule of the nephron consists of proximal convoluted tubule. See, this is the proximal convoluted tubule. Why it is called as proximal? Proximal means anterior part. So this is the anterior region of the tubular part of nephron. Okay, and this end which is there of the proximal convoluted tubule is open end. Got it? So proximal convoluted क्यों बोला है? Because it is coiled. Proximal because it is near to the Bowman's capsule. Convoluted because it is coiled. Okay, so proximal convoluted tubule, very first part of the renal tubule of the nephron. Then second part of the renal tubule of the nephron is loop of Henle's. So this is the loop which is formed. Hairpin loop like structure which is formed here of the nephron. Okay, then third part of the renal tubule of nephron is distal convoluted tubule. So distal because it is away from the Bowman's capsule. Convoluted, convoluted क्यों बोला है? Because again it is coiled, coiled tubular part. So tell me how many parts are there of the renal tubule of the nephron? Three. Proximal convoluted tubule, distal proximal convoluted tubule, loop of Henle's and distal convoluted tubule. Okay. Now this distal convoluted tubule of the nephron is opening in the collecting duct. Okay. So how is the collecting duct? Again it is tubular structure. Collecting duct is bigger and it is receiving many distal convoluted tubules of many nephrons. Okay, so one collecting duct will receive 8 distal convoluted tubule of different nephrons. Okay, so total 8 nephrons के distal convoluted tubules यहाँ पे एक collecting duct में open होते हैं. Okay, so look here, 1, 2, 3, 4, 5, 6, 7 and 8. Okay, so 1, 2, 3, 4, 5, 6, 7, 8. Total 8 distal convoluted tubules of 8 different nephrons are opening in one collecting duct. Likewise, millions of nephrons are present in each kidney. So many collecting ducts will be opening in the common duct, that is duct of Bellini. सो बहुत सारे ऐसे कलेक्टिंग डक्ट्स रहेंगे किडनी में which is going to open in the duct of Bellini, and this duct of Bellini will then open in the minor calyx through renal papilla. So this is the renal papilla, and through renal papilla this urine will enter in the minor calyx. Okay, here I have drawn LS of kidney. You all know cortex region and medulla region. What I told you, which is the renal papilla? This is the renal papilla. Okay, so renal papilla is receiving which duct? Duct of Bellini. How duct of Bellini is formed? Due to union of many collecting ducts. So many collecting ducts are opening in duct of Bellini, and duct of Bellini is opening in renal pelvis. Renal pelvis is opening in minor calyx. Many minor calyx unite to form major calyx, and major calyx will open in the renal pelvis. Renal pelvis will further open in the ureter, and likewise then urine will be stored in the urinary bladder for some time, and afterwards it will be released out of the body through urethra. Got it? So this is how actually conduction of the urine will take place in the nephron. Okay, so nephron, which is there, why it is known as functional unit of kidney? Because it is responsible for formation of urine. It brings about selective reabsorption as well as tubular secretion, and it also removes the unwanted products out of the body. जो excess है वो भी remove करता है और nitrogenous waste भी remove करता है. Okay, so which are the two parts which we have studied of the nephron? Nephron consists of Malpighian body and renal tubule. Okay, Malpighian body which is there, it consists of Bowman's capsule and glomerulus. Okay, Bowman's capsule is cup-shaped structure, glomerulus is network of blood capillaries. How they are formed? Due to entry of the afferent arteriole, which will divide and redivide to form network of blood capillaries, that is glomerulus. Glomerulus will, blood capillaries of the glomerulus will rejoin to form efferent arteriole. Efferent arteriole will come out from the Bowman's capsule of the nephron, and it will divide and redivide into many peritubular capillaries. These peritubular capillaries will surround the nephron. Okay, and likewise then there will be urine formation. Similar way, then next part of the nephron which I told you, first part Malpighian body, second part is the renal tubule. Renal tubule consists of proximal convoluted tubule, loop of Henle's which is hairpin like, and distal convoluted tubule and collecting duct. Okay, so three regions are there of the renal tubule: proximal convoluted tubule, loop of Henle's and distal convoluted tubule. Distal convoluted tubule, 8 distal convoluted tubules of 8 different nephrons will open in the collecting duct. Collecting duct of many nephrons will open finally in one common duct, that is duct of Bellini. Duct of Bellini will open in the minor calyxes through renal papilla. Got it? So now we will study what are the different cells which are present in this nephron. Okay, so कौन से cells रहते हैं? Here in the Bowman's capsule lining, this is the outer lining and this is the inner lining. Outer lining of the Bowman's capsule is known as parietal layer, and inner lining is known as visceral layer. Okay, so parietal layer as well as visceral layer are made up of squamous epithelial cells. They are made up of squamous epithelial cells. Proximal convoluted tubule, it is made up of cuboidal epithelium. Like this, cuboidal epithelium में क्या रहता है, कैसे रहते हैं cells? Cube like, having rounded nucleus in the center, and this cuboidal epithelial cells are having microvilli. So these microvilli will help in absorption of nutrients. Okay, nutrients, then sodium ions, potassium ions, calcium ions will be absorbed by these cuboidal microvilli cells. So brush bordered cuboidal epithelium will be seen in the inner lining of the proximal convoluted tubule, which will help in selective reabsorption. Selective क्यों बोला है? Because our body will select which all salts, which all ions has to be absorbed. Okay, then nutrients, glucose, amino acids तो लगता ही है body को, तो वो reabsorb किया जाएगा. First absorption where it will take place? After digestion, absorption will take place in small intestine, and these nutrients will enter in the blood, and few of the nutrients may again enter in the nephric filtrate because of this pressure difference. So whatever important nutrients are there which has to be again reabsorbed. So इसीलिए उसको क्या बोला है? Selective reabsorption. So selective reabsorption is the main function of the proximal convoluted tubule of the nephron. Okay, no doubt other tubules of the nephron are also involved in reabsorption, but major reabsorption कौन करता है? Proximal convoluted tubule, because proximal convoluted tubule is having microvilli cuboidal epithelial cells. So brush bordered cuboidal epithelial cells are present in the inner lining of the proximal convoluted tubule. Okay, now coming to the next part of the renal tubule of nephron, that is loop of Henle's. Okay, so loop of Henle's which is there, it consists of hairpin like structure. So look here, it is just like the hairpin which we put in our hair. Okay, U shape है, ये straight है. How many limbs are seen here of the loop of Henle's? This is the descending limb. नीचे की तरफ जा रहा है descending limb, and this is the ascending limb. Descending limb which is there, it is having thick part and it is also having thin part. Thick descending limb is very less as compared to thin descending limb, and thin descending limb, whole descending limb which is there, it is responsible for reabsorption of water. Okay, it is permeable to water. So water can pass in the blood from this tubule, but it is impermeable to salts. NaCl नहीं पास हो सकता है tubule में से blood के अंदर. Okay, so Na positive, Cl negative, NaCl will not enter in the blood through through descending limb. Okay, so descending limb will only be responsible for reabsorption of water. So it is permeable to water but impermeable to salts. Okay, coming to the another limb of the loop of Henle's, that is ascending limb. Ascending limb which is there, it is having some part which is thin and some part which is thick. Okay, so thick part ज़्यादा रहता है, distal descending limb में thick part कम रहता है, thin part ज़्यादा रहता है. Ascending limb में thin part is less, thick part is more. Okay, ascending limb which is there, it is impermeable to water but permeable to salts, that is NaCl. So NaCl can easily enter from the nephric filtrate in the blood through ascending limb. So very important question which is asked in exam. Descending limb is permeable to water, impermeable to salt, wherein ascending limb is permeable to salt, NaCl, Na positive as well as Cl negative ions, and impermeable to water. Okay, then coming to the cells which are present in the descending limb and ascending limb. Cells कौन से रहता है descending and ascending limb में? Squamous epithelial cells and cuboidal epithelial cells. Squamous and cuboidal epithelial cells. Okay, now coming to the next part of the renal tubule, that is distal convoluted tubule. Distal convoluted tubule, distal क्यों बोला है? Because it is away from the Bowman's capsule and it is consisting of cuboidal epithelial cells, and here these cuboidal epithelial cells will have few microvilli. Okay, सबसे ज़्यादा microvilli कौन से cuboidal cells में रहता है? Proximal convoluted tubule के cells में रहता है. Got it? Because maximum selective reabsorption takes place in the proximal convoluted tubule. So distal convoluted tubule is lined by cuboidal epithelial cells, and these cuboidal epithelial cells have few microvilli. Okay. Now coming to the collecting duct. Collecting duct will have squamous epithelial cells and few of the cuboidal epithelial cells. Got it? So now here I have told you efferent arteriole will divide and redivide into many blood capillaries, and it will form peritubular capillaries. So look at the diagram. I have drawn this division of the efferent arteriole into many blood capillaries. Okay, so the blood capillaries, few blood capillaries will surround the loop of Henle's in U shape. Okay, so U shape में वो surround कर रहा है, इसीलिए वैसे वाले स्ट्रक्चर को क्या बोलेंगे? वासा रेक्टा. Okay, so this is the vasa recta. So vasa recta is the blood vessels or blood capillaries surrounding the loop of Henle's, and this vasa recta is also having the blood, and it is also having the U shape like the loop of Henle's. So loop of Henle's का U shape है, वैसे ही vasa recta का भी U shape है. But the direction of the nephric filtrate and direction of the blood is opposite. Nephric filtrate कैसे move हो रहा है? ऐसे move हो रहा है, from PCT to loop of Henle's to DCT, wherein how is the direction of the blood flow in the vasa recta? It is opposite. अभी देखो nephric filtrate ऐसे flow हो रहा है descending limb से, wherein the blood which is flowing in the vasa recta is in upward direction. Ascending limb में nephric filtrate is moving in this direction and blood is moving in opposite direction in the vasa recta, which is present next to the ascending limb of the nephron. Got it? So very important, you have to remember the direction and what is vasa recta. Vasa recta is the U-shaped blood capillaries which are present around the U-shaped loop of Henle's. Okay. Now looking at the overall structure of the nephron, you can make out which is the part of the nephron which is seen in renal cortex and which is seen in renal medulla. I have made partition indicating dotted line here, renal cortex region of the kidney and renal medulla region of the kidney. So renal cortex में कौन कौन सा part आ रहा है नेफ्रॉन का? Malpighian body, proximal convoluted tubule and distal convoluted tubule, and some part of the collecting duct. So renal cortex means this part. Renal cortex dark red color का क्यों है? Because it is having Malpighian body, convoluted part of the proximal convoluted tubule and convoluted part of the DCT and some part of collecting duct. Okay, wherein renal medulla is pale red in color. Why it is pale red in color? Because it is not having any coiled structure as well as it is not having the glomerulus. Okay, जिसकी वजह से pale red color आएगा उसको. So what you can see here, which part of the nephron will be there? Renal medulla में मैंने क्या दिखाया है? ये region में loop of Henle's is there, and next part, posterior part of the collecting duct. So ये region, that's why we are drawing many striations in the medulla because of loop of Henle's and collecting duct. Okay, so we have studied in this particular session structure of nephron. Okay, in structure of nephron we have studied two major parts of the structure of nephron. First is Malpighian body and second is renal tubule. Okay, thank you.